You know, the tech that's going to power our 6G world, it's already in the works. But here's a thought. What if the very waves that connect all our devices are also changing our cells in ways we just don't understand yet? Today, we're diving into a really fascinating new study that looks past the obvious and into the invisible dance between technology and our own biology.
I mean, we're all pretty used to 5G now, right? Streaming, connecting, everything's faster than ever. But you know how it is in the tech world. The next big thing is always just around the corner. So yeah, what does come after 5G?
Well, the answer, if you ask the engineers and scientists, is all about terahertz waves, or THz for short. Think of it as this slice of the electromagnetic spectrum that's kind of sandwiched right between microwaves and infrared light. And the promise here is huge. We're talking about powering everything from ridiculously fast 6G networks to brand new kinds of medical imaging and super advanced motion sensors.
But you know, with any new tech, there's always that big question. Is it safe? I mean, sure, we know that high-power THz waves can heat things up. That's a given. But the real mystery, the thing scientists are really scratching their heads about, is whether these waves can have, well, non-thermal effects. In other words, can they mess with our cells without raising their temperature even a tiny bit? That is the puzzle we're digging into today.
Okay, so here's our game plan. We'll start with the terahertz puzzle itself. Then, we'll zoom right into the living cell membrane. We'll check out the super clever experiment they ran, reveal this kind of surprising melting effect they found, and see what water has to do with all of it. And finally, we'll wrap up with what this all means for the future.
All right, let's get into it. The terahertz puzzle. For a long time, the thinking was pretty simple. If THz waves aren't powerful enough to actually heat up our cells, then they don't do anything. End of story. But you know, good science is all about spotting the weird stuff. And some really strange results started popping up in labs. Things like certain THz frequencies speeding up how proteins form, or even messing with cell division, all without any noticeable change in temperature. Something else was definitely going on.
So to try and solve this thing, the researchers behind this study had to pick a target. And they zeroed in on one of the most important parts of any cell, its membrane. You can really think of it as the cell's bouncer or gatekeeper. It's the barrier that keeps the organized stuff inside safe from all the chaos on the outside. So picture this. The cell membrane is like a super flexible, kind of oily bubble, and it's made of two layers of these molecules called lipids. But, and this is the part you really need to remember, the outer surface of this bubble absolutely loves water. It's always interacting with the water molecules all around it, forming these strong connections called hydrogen bonds. And trust me, this relationship with water, it's going to be a huge piece of the puzzle later on.
Okay, so here's the tricky part. How on earth do you measure a change in a cell membrane if it has nothing to do with heat? I mean, that's the core challenge, right? The scientists had to figure out a way to completely separate any non-thermal effects from the simple heating ones.
Their secret weapon was a technique with, well, a bit of a mouthful of a name, fluorescence recovery after photobleaching. We'll just call it FRAP. It's actually a super clever way to measure how fluid, or kind of runny, a cell membrane is. The basic idea is you tag the membrane with these glowing dyes, then you zap one little spot with a laser to turn the lights off there, and then you just watch and see how fast the other glowing molecules rush in to fill that dark spot. And this diagram here shows you exactly what I'm talking about. So, in step 1, you see the whole membrane is glowing. Then, step 2, bam, a laser zaps a tiny area, and now you've got a dark spot. And then, step 3, that's the magic. We just watch as those glowing molecules from the outside move in and fill that dark patch back up. The faster that spot disappears, the more fluid, or you could say more disordered, the membrane is. It's a really neat way to get a hard number on the membrane's condition. Now, this next part is the absolute key to the whole experiment. To be 100% sure they were only seeing non-thermal effects, the researchers used this ridiculously sensitive, super thin device to measure the cell's temperature with incredible precision. They basically calibrated everything so perfectly that any heat from the THz waves was canceled out. This way, they could be sure temperature wasn't a factor at all in what they saw. So, the stage is set, right? The experiment is perfectly controlled. So what happened when they finally turned on the terahertz waves?
Okay, this graph right here, this is the core finding. Each one of those dots represents how fluid the membrane is at a certain temperature. See the black squares? That's our baseline, no THz waves involved. Now look at the other shapes, those are two different THz frequencies. See how below about 36 degrees Celsius, those points are way higher than the black squares? That is the smoking gun.
It means the membrane got way more fluid when the THz waves were on. But, and this is key, only at these cooler temperatures.
So let's just break that down real quick. First finding. Certain THz frequencies definitely made the cell membrane more fluid. Second, this effect only kicked in at cooler temperatures, below the normal cell growth temp. Third, and this is the big one, we know for sure this isn't a heat effect because they controlled for that so carefully. And, super interesting, another, higher frequency they tested did absolutely nothing. So this is a really specific interaction we're seeing.
Of course, good scientists always double-check their work, right? So they ran a whole other experiment using a special dye that also measures how orderly a membrane is. And guess what? The results lined up perfectly. At the cooler temp, 30 degrees, the THz waves made the membrane way more disordered. But up at 40 degrees, no difference at all, just like they saw before.
The researchers themselves came up with a great way to describe it. They said the THz waves essentially melt the nice, ordered, almost gel-like structure of the cell membrane. It turns it more fluid, but only when the membrane is in that cooled, ordered state in the first place.
Okay, but how? How can a wave melt something without any heat? Well, the answer seems to come back to that one little detail I told you to remember: water. Take a look at this diagram. It shows their best guess for what's happening. On the left, you've got that cool, orderly membrane. The theory is that the THz waves aren't hitting the membrane lipids directly. No, instead, they're zapping and energizing the water molecules that are stuck to the surface. This makes the water molecules jiggle around and gives them just enough of a kick to actually push their way into that oily lipid layer, and once they're inside, they totally mess up that neat, orderly packing, which causes the whole thing to switch to that more messy, fluid state you see on the right.
So look, this is all really cool science. But what does it actually mean for us in the real world? Why should we care if a cell membrane gets a little more fluid?
Well, it's pretty much a classic double-edged sword. On the one hand, this is a challenge. It's telling us that our safety standards for new tech can't just be about heat. We've got to start thinking about these sneakier, non-thermal effects. But on the other hand, this opens up a massive opportunity. I mean, if we can learn to use these waves to control a cell's membrane on command, we could develop some unbelievable new medical treatments. Think about making it easier to get life-saving drugs directly inside a cell. The possibilities are huge.
So what this study gives us is the first real, hard evidence of a non-thermal effect, plus a pretty convincing idea of how it works. It's a powerful reminder that even with the tech we use every single day, there's this incredibly complex, invisible world of stuff happening at the cellular level. And it leaves us with this one big question to think about. As we keep pushing the boundaries of technology, what other invisible interactions are just waiting for us to discover?